At the beginning of the 20th century, one of the most important scientists in the United States was Charles Doolittle Walcott. At various times, Walcott was the director of the U.S. Geological Survey, secretary of the Smithsonian, and president of the American Association for the Advancement of Science, the Geological Society of America, and the National Academy of Science, three of the most important scientific organizations in the country. He even advised President Theodore Roosevelt on science matters. Walcott was born in New York in 1850. Although he developed an interest in nature at a young age, he never completed high school. Instead, he focused his attention on locating and selling fossils as a commercial fossil collector. Through his work, Walcott launched a long career in geology and paleontology. In 1886, while working for the U.S. Geological Survey, Walcott received a small collection of fossils from Mount Stephen, a mountain located in the Canadian Rockies of British Columbia. It is located in an area that is now known as Yoho National Park. Walcott studied the fossils and wrote several papers about them. All the while, he became increasingly interested in the geology and paleontology of the Canadian Rockies. Between 1907 and 1925, Walcott spent almost every summer in the Canadian Rockies, exploring the regional geology and amassing an enormous collection of fossils. He often brought his family with him when he did his fieldwork. They included wife Helena, daughter Helen, and sons Sidney, Benjamin, and Charles. In late August of 1909, at the end of his field season, Walcott was riding his horse over the rocks of Mount Burgess, just a few kilometers north of Mount Stephen. On his ride, Walcott made a momentous discovery. Along the trail, he stumbled across, quote-unquote, many interesting fossils of animals. They were unlike any that he nor anyone else had ever seen before. These fossils, he knew, dated back to the Middle Cambrian period. Since then, we've learned that the fossils are about 508 million years old. Walcott found an abundance of fossils. They all occur as black compressions which have been flattened between layers of fine-grained black shale. But what really amazed Walcott was the wide array of forms and shapes he found among the fossils. They were truly unlike anything he'd ever seen before. Each one, it seemed, was completely unlike the last. Over the next few days, Walcott and his family would collect many fossils, which he describes here in his field notebook, dated between August 31st and September 3rd of 1909. Despite his immediate excitement for his discovery, which we now call the Burgess Shale, it came too late in the summer for Walcott to mount a serious expedition. Nonetheless, Walcott returned the following summer in 1910 with his family to begin an extensive excavation of the site. You can see them here at Wapta Falls, not too far from the Burgess Shale, in July of 1910. Their journey began in Washington, D.C., where they made their home, and took them across Canada on the Grand Trunk Pacific Railroad. The train took them to the town of Field, which today has a small population of around 169 people. It is located within the confines of Yoho National Park. There, they mounted their expedition out of Mount Stephen House, located near the base of Mount Stephen. In those days, the country was rough, and there were few automobiles available for travel. So they returned sparingly to Field. Instead, the entire family, including daughter Helen, shown here in 1910, made the journey to the Burgess Shale on horseback. The family established a base camp for their work at Burgess Pass, not too far from the actual site of the fossils. There, they set up tents, equipment, and other instruments needed for their survival and research. Higher up on the steep slope of Mount Burgess, against the backdrop of Wapta Mountain, the family began to excavate the Black Shale, opening up a small quarry in the process. The quarry is now known as the Walcott Quarry. Excavating rocks from the quarry was tedious work in 1910, just as it is now. Walcott and his sons would break the shale into large pieces. They then slid the useless blocks, the rocks with no fossils, down the slope of the mountain. They carried the rest of the rocks, the good pieces, back to their camp at Burgess Pass. There, the family would use hammers and chisels to carefully split the rocks open, just as you see Charles and Helena doing here. When they ultimately found fossils, the Walcotts, like Stewart here, would pack the fossils and send them back to Mount Stephen House in Field, so they could be shipped back to their home in Washington, D.C. Over the next 14 years, Walcott and his family made many trips to the Burgess Shale. The sheer volume of rock that they explored was massive. In the process, they amassed a staggering number of specimens, over 65,000 fossils in total. Most of the fossils were ultimately deposited at the Smithsonian Institution. Of course, when he wasn't in the field, Walcott spent a great deal of time studying and describing the fossils. In 1911 alone, he published his research on the Burgess Shale in 3 separate volumes of the Smithsonian Miscellaneous Collections. These publications introduced the world to some of the most bizarre creatures that have ever existed on our planet. In addition to trilobites and many other strange-looking arthropods like Morella and Opabinia, Walcott also described sponges, algae, brachiopods, mollusks, and an outstanding array of worm-like organisms. There were also many organisms that he could not easily classify. Indeed, because the fossils were so strange and unusual, we now know that he misclassified a great many of them. For example, he originally described specimens like this one as jellyfish, when in fact they are the mouth parts of an arthropod organism called Anomalocaris. Despite these issues, the discovery of the Burgess Shale in 1909 did receive some national attention and publicity. Walcott described his experience working on it in the National Geographic magazine in 1911. Nonetheless, scientists generally thought of the fossils as simply bizarre curiosities, nothing more. They would not begin to really appreciate their value for another half century, long after the time of Walcott. Walcott made his last trip to the Burgess Shale in 1924 at the age of 74. He then passed away 3 years later at the age of 76. It is safe to say that Walcott left behind an impressive legacy. However, despite all his hard work, talent, and good fortune, Walcott was not perfect. He overlooked something very, very important about the Burgess Shale. Walcott incorrectly believed that all of the fossils in the Burgess Shale could be classified into living taxa. In other words, Walcott believed that all of the species in the Burgess Shale belonged to crown groups rather than stem groups. It is helpful to think about crown groups and stem groups in terms of a phylogenetic tree like this one. A crown group includes a species and all of its descendants, both living and extinct. This means that a crown group includes all extant species and their last common ancestor. Below crown groups in a phylogenetic tree, you find stem groups. Stem groups consist exclusively of extinct taxa, which have some but not all of the characters of modern species. Let's look at a real example. Arthropoda is the most diverse phylum of animals on Earth today. It includes insects, crustaceans, horseshoe crabs, spiders, and scorpions. Because all of these taxa are alive today, they belong to the crown group of arthropods. Crown group arthropods all have 4 characters in common. They all produce chitinous exoskeletons. They all periodically molt these exoskeletons over the course of their lifetimes, and they all have segmented bodies and segmented appendages. You find these characters in all crown group arthropods, including trilobites, which went extinct over 250 million years ago. Of course, these characters did not all evolve at the same time. Some of the ancestors of arthropods had some, but not all of these characters. There may have been extinct arthropods that produced and molted exoskeletons with segmented bodies, but did not have segmented appendages. We refer to these extinct arthropods as stem group arthropods. They possessed some, but not all, of the characters you see in the crown group. Walcott incorrectly believed that all of the species in the Burgess Shale belonged to crown groups. He did not appreciate that many, and perhaps most, of the species in the Burgess Shale actually belonged to stem groups, not crown groups. History would prove that Walcott made a significant oversight. It wasn't until the 1960s, 70s, and 80s that a new generation of scientists would revisit the Burgess Shales and dedicate themselves to studying and understanding it in all its glory. This generation included Alberto Simonetta, Harry Whittington, Desmond Collins, Derek Briggs, and Simon Conway Morris, among many others. This new generation returned to the Walcott Quarry. There, they jump-started a new period of excavation which has been ongoing for most of the past few decades. They also began to discover new sites and open up additional quarries, like the Raymond Quarry shown here. Altogether, this work resulted in the recovery of an astounding number of fossils, most of which is managed by the Royal Ontario Museum in Field, British Columbia. Today, the Burgess Shale is considered a UNESCO World Heritage Site, making it one of the most protected and cherished natural landmarks in the world. Visitors are allowed to visit the site in small groups, but fossil collection is not permitted except under the most extreme circumstances and requires special permission and permits, which are hard to come by. Violation of the conservation laws are met with steep penalties. A hiker was recently fined $4,000 for taking just one small fossil. So why exactly is the Burgess Shale so special? From my perspective, there are three reasons. First, the fossils have an amazing amount of detail. The Burgess Shale is an exquisite example of a conservat Lagerstätte, a geologic deposit or storage place for exceptionally preserved fossils. And trust me when I tell you, there are few better Lagerstätten in the entire world. It can be hard to appreciate at times, but the preservation of the fossils is truly exceptional. Fossils have features that you won't find anywhere else, including in other Lagerstätten. Fossils contain remains of eyes, gut contents, muscles, and many other delicate features. Second, the Burgess Shale provides an incredible snapshot of a community of marine animals that lived in the ocean during the Cambrian period. Outside of Lagerstätten, most fossils are hard parts, like shells. Although these hard parts are very abundant in the fossil record, they aren't representative of the real world. They provide highly incomplete and biased snapshots of the ancient world. For the Cambrian, this means we ordinarily find only trilobites, brachiopods, and mollusks. But the fossils of the Burgess Shale fill in the picture. They create a complete picture of the community as it was 508 million years ago. They provide evidence for a rich and complex community of marine organisms that lived during the Cambrian period. They show us an ocean filled with arthropods, sponges, algae, cnidarians, and even some chordates and hemichordates. Not surprisingly, these fossils have inspired a significant amount of research on the paleoecology of the Cambrian Ocean. How many species were there? Which type of animals were most abundant? How many species were nectonic? How many were benthic? How many species were predators? How many were scavengers? How many were algae? And how did they all interact with one another? The final, and I think most important reason that scientists value the Burgess Shale today is perhaps the hardest to appreciate if you are someone who is still learning about the diversity of life on Earth. The taxa of the Burgess Shale are unique and diverse. They exhibit incredibly high morphological disparity for animals of any age. We know for a fact all of the Burgess Shale fossils shown here are animals, but many of them are unlike any animals that we find on Earth today. Take, for example, this fossil. Charles Walcott found many fossils like this one, which he interpreted as a polychaete worm, which are common in the modern ocean. However, there were some scientists who disagreed with him. In 1977, a graduate student named Simon Conway Morris, now a professor at Cambridge, revisited this taxon. He was reasonably certain that the creature was some sort of worm-like animal, but he was mystified by its bizarre characteristics. It presented a significant challenge. Look carefully. What is the head and what is the tail? Put another way, which side is up? There were two possibilities, and the correct answer wasn't immediately clear. For this reason, Conway Morris gave the genus an unusual name. He called it Hallucigenia, as in, if you stare at it too long, you may begin to hallucinate. Simon Conway Morris interpreted Hallucigenia this way, identifying the long features as legs and the short ones as features located on its back. However, we've since come to learn that Simon Conway Morris had it backwards, or should I say, upside down. We now know that Hallucigenia was a worm-like animal, one with long spines on its back. These spines may have protected it from predators. What we don't know is where it belongs on the phylogenetic tree of animal life. What sort of animal was it exactly? All we know for sure is that it was a weird one. But it was not alone. Indeed, many of the worm-like animals in the Burgess Shale appear to have been covered in armor or spikes, things you simply don't find in modern worms. This is Waxia, another odd creature. Its entire body was covered in scales and spines. Waxia may have been a worm, or alternatively, it may have been a primitive mollusk, a relative of bivalves, snails, and cephalopods. It is difficult to be sure when the fossils look so alien. In any case, these scales and spines also likely protected Waxia from predation of some kind. Speaking of predation, say hello to Anomalocaris. This creature may have been one of the first great predators on our planet. It is generally thought that it was the largest predator in the Burgess Shale community. Anomalocaris grew up to 1 meter in length and probably swam through the water with great speed by propelling itself with undulating flaps or fins located at the sides of its body. It may have fed on arthropods, worms, or other critters located on the seafloor. Anomalocaris had a segmented body, so paleontologists are reasonably certain that it was an arthropod of some kind. But it doesn't resemble any crustaceans, horseshoe crabs, or other arthropods that are alive today. In all likelihood, Anomalocaris is one of many stem group arthropods in the Burgess Shale. Indeed, most species in the Burgess Shale may be stem group arthropods. Between the exceptional preservation of its fossils, the completeness of its paleo community, and the vastness of its taxonomic diversity and morphological disparity, the Burgess Shale provides one of our best windows into the past, granting us access to study life at the height of the Cambrian explosion. It paints a portrait of a wild and ever-changing ocean, one entirely unlike the world as it is today. Marine animals, for the most part, were every bit as morphologically complex and ecologically diverse as they are today. But the Cambrian was a tumultuous time of evolution and experimentation. Over a short span of time, maybe 10 to 20 million years, the Earth saw the rise and fall of many new forms of animal life. Some taxa, like Hallucigenia, existed for a brief time before going extinct. They are now simply footnotes in the history of life. Other taxa survived the Great Experiment and went on to dominate the planet, becoming the crown groups on the tree of life. Regardless of why some taxa survived the Great Experiment and others did not, one thing must be true. The Cambrian explosion had a long-lasting impact on the evolutionary trajectory of life on Earth, setting the stage for all things to come. Today, we know that the Burgess Shale is not unique. In fact, we now know that similar Lagerstätten and storage places occur in rocks of Cambrian age all around the world, most notably in China, Australia, Greenland, and the Great Basin in the US. But the Burgess Shale remains the first and arguably the best of the bunch. Although one could argue that Charles Walcott did not fully comprehend the implications of his discovery, it is clear that he did appreciate its value. His extensive fossil collections, many publications, and contributions to paleontology and geology as a whole continue to reverberate throughout the scientific community.